Bismillahirrahmanirrahim. I am Dr. Huma Naz. I am working as assistant professor in Cholisan Institute of Biological Sciences, US Bahawalpur. In this topic, I will explain how glycoprotein like mucin isolated from fish skin mucus, and this work is a part of my NRPU project entitled Exploring the Clues of Fish Skin Mucus Against Pathogenic Microbes. First of all, what are the glycoproteins? Glycoproteins are the proteins that have one or more carbohydrate chains covalently attached to them. These carbohydrates can be simple sugar or more complex oligosaccharides. Glycoproteins are found in all living organisms and play a wide variety of roles in cellular functions, including cell recognition, signaling, and structural support. Fish mucus contains glycoproteins, specifically mucins, which are large, heavily glycosylated proteins responsible for the mucus viscosity and have protective properties. These glycoproteins play a crucial role in the innate immune system of fish, acting as a physical and chemical barrier against pathogens and pollutants. They also contribute to lubrication and sensory functions. Here I will explain the methodology for glycoprotein isolation. To isolate the glycoprotein, the following steps were followed. First of all, fish mucus of 3 fish species was gently isolated by scrubbing using a plastic spatula. Next, dilution of mucus 4 times with distilled water. After that, the obtained gel was homogenized and dialyzed against distilled water using a dialysis membrane. This dialysate was finally centrifuged. After centrifugation, gel filtration chromatography was performed for further purification of glycoprotein. To obtain the elutions, phosphate buffer was used, and after obtaining 30 elutions, the absorption of each elution was measured at 250 nanometer and 218 nanometer. After that, PSSA was used or performed to identify the glycoprotein in the purified samples, and after identification, the absorbance was measured at 550 nanometers to identify the concentration of glycoprotein in each sample. Here is the flow chart of methodology which was followed for the purification or isolation and purification of glycoprotein. Results. The results obtained from the purification of mucus or purification of glycoprotein from 3 fish species show that the 30 fractions were taken from each fish species. The absorbance of each fraction or elution was noted at 280 nanometer and 215 nanometer. The 3 fish species Oreochromis niloticus, Labiorhita, and Dinofrangdon adila was used for this experiment, and from these 3 fish species, the glycoprotein was isolated from skin mucus of these 3 fish species. This is a table which is showed the results of gel filtration chromatography. The yellow highlighted values shows that these fractions may have highest concentration of glycoprotein, and the fractions were further used for and these yellow colored values, the fractions were further used for PSSA to identify the type of glycoprotein. And these graphs are also shows the purification summary of glycoprotein. After obtaining highest values, these highest fractions were used for periodic acid chef assay, which detect the uh, uh, glycoprotein by targeting their carbohydrate group. Periodic acid oxidizes the sugars, enable them to react with chef reagent. A visible magenta color confirms the presence of glycoprotein. PSSA was performed on the highest fractions from three fish species obtained from gel filtration after gel filtration chromatography technique at 250 nanometer and 280 nanometer. The samples from each fractions to 25 microliter were incubated with 120 microliter of periodic acid in a 7% acetic acid for 90 minutes, followed by 100 microliter chef stain for 60 minutes at room temperature. The development of purple color confirmed the presence of neutral mucin in the skin mucus of three species. Here is a sample on which the PSFA was performed, and the magenta color of these samples shows that all these three fish species have glycoprotein, and this glycoprotein is a this glycoprotein is a mucin, and mucin is a and in mucin it's not a acidic. This color show that it is a neutral mucin. And the conclusion of this study is that we have successfully isolated and verified the mucin glycoprotein from fish skin mucus. Neutral mucin was confirmed by PSSA, and the finding of this work provide a foundation for further for future research on natural bioactive compounds in aquaculture and in biomedical applications. Thank you.